Okay, so now we will be discussing cell structure and function. So basically in the first episode of the biology series, I did cells, basically plants versus animal cells, and just some characteristics of living organisms. Now we're going into more specifics about the cell structure and the function of the cell, basically, right? So let us begin. So we need to talk about what are cells first. Like in the first video, we mentioned some of this already, but let us mention it all over again. So basically, cells are the basic units of life, right? So they're the basic units of life, and or what am I saying? Okay, organelles are the components that make up cells, basically, right? So I was going to mention the cell tenets again, right? So there are three tenets of the cell theory that basically states that all cells are made up of living things, all cells are made from pre-existing cells, and cells are the basic unit of life. So basically this is where this definition comes from, where it says that cells are the basic units of life. So everyone is made up of cells, me, you, the little girl, everybody is made up of cells. So within our cells, we have some structures called organelles. So organelles are the components that are basically in our cells. So like our nucleus, our mitochondria, chloroplast, just any structure within the cell, vacuoles, all of those things are considered as cell organelles basically. And basically cells were discovered by a scientist named Robert Hooke, right? So basically it's a lot that goes into cells overall, as you can see. So you need to remember that cells are the basic units of life. Organelles are the components that make up cells. So example are nucleus, chloroplast, vacuoles, endoplasmic reticulum, all of those things are cell organelles. And then cells were discovered by a scientist named Robert Hooke. It's just a fact, just to know it basically, right? So let's talk about the functions of the cell. So cells have a lot of functions. They have a lot of functions, like they're the basic units of life overall, literally. So imagine something making up me, you, and everything, and cells are literally so small. They are like really, 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 really small. And in just one person's body, there are probably billions of cells. At this moment, we don't remember the exact amount of cells that's in the human body, like the approximate, we don't remember it right now, but we have a lot of cells in our bodies. So they have a lot of functions, right? So they play a structural role, where we say that cells provide structure and support for the body, because without cells, where would the body even be? Because they are like the little building blocks that make up all living organisms, not just human beings, not just cats and dogs. Literally, the fish we swim in the water, literally, the algae, literally anything that carries out the 7 characteristics of life, as long as it grows, as long as it can reproduce, as long as it can respond to stimuli, it has like irritability, as long as it can carry out some form of movement, some form of feeding slash nutrition, some kind of excretion, and some kind of respiration, then it has cells, because that is when we would consider it to be living, right? So that cells basically also facilitate growth through mitosis. And we know that mitosis basically division of cells to produce two daughter cells in the end. And then we have energy production. Cells convert nutrients from food into energy, and we know which organelle is responsible for energy production. That would be our mitochondria. That plays an absolute big, like a really, 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 really big role in energy production within our cells, right? So the mitochondria, the powerhouse. I think that was the first like definition I learned about mitochondria, that it was the powerhouse basically. So then it plays a role in transport. So cells import nutrients and exports waste products. So So basically these functions right here, each organelle literally plays a role in each of these functions, right? So it's the organelles within the cells that allow all of these things to basically take place essentially, right? Because literally as I'm going through the list, the name of the organelle is coming directly to my mind. So each organelle is responsible for something here. So we said that metabolism, cells perform chemical reactions that keep the organism alive. Reproduction, cells help with reproduction through mitosis and meiosis. And mitosis and meiosis are they're basically the two ways by which reproduction happens. We know mitosis happens in somatic cells, meaning body cells, and we know that we produce two daughter cells in the end, and the meiosis is basically includes the fusion of gametes. So this is like the sexual aspect of reproduction, right? So mitosis is more, I would say, kind of asexual, right? Because it's body cells. So there is no fusion of gametes or anything of that sort. But in our meiosis, we have to have some fusion of gametes. We're going to need a male and a female in order for some sexual reproduction to take place in meiosis. And we know that we get haploid daughters in the end. So four haploid daughter cells, right? So now communication. So cells communicate with each other and the environment through receptors, right? So detoxification. Some organelles like the smooth endoplasmic reticulum detoxify the cell, meaning that they get rid of toxins. So let us get into more detail regarding each cell organelle. Alright, so that was just like a little summary. Let's get into more details. So before we get into that, we have to know that there are two main broad categories that all cells fall into basically. So it's either the cell is a eukaryotic cell or the cell is a prokaryotic cell. So for our eukaryotes, they have nucleuses, right? So once you hear eukaryotes or eukaryotic, it means true nucleus. So it means that these cells have nucleus, right? And prokaryotes, they don't have nucleuses, right? So that would be like our bacteria. And there's another term here, archaea, which you don't really have to know about archaea at this level, but it's there. It falls under that category of prokaryotic cells. So we, as human beings, we are made up of eukaryotic cells. So basically, true nucleus, right? So eukaryotic cells are basically in organisms, right? Are organisms that have membrane bound nucleus. We have membrane bound organelles within our body. So we have our nucleus, we have a mitochondria, we have well, not we, but the plants have chloroplast, we have our vacuoles. So all of these membrane bound organelles, we have them because we have eukaryotic cells. In the case of bacteria, in the case of prokaryotic cells, those cells do not have a nucleus. They do not have membrane bound organelles. They don't have it. They don't have it. So those don't have membrane bound organelles. They don't even have a true nucleus. They don't. They really don't, right? They're a lot more simpler in structure if you want to say that. But yes, they don't have any membrane bound organelles. They don't have a true nucleus. And basically our prokaryotes have both a plasmid and a nucleoid, right? So the nucleoid has most of its genetic information. So instead of having a nucleus, it has a nucleoid. And then we also have the circular DNA molecule, which is floating freely in the cytoplasm, which is called a plasmid basically. So that's our prokaryotes. Okay, so let's do a quick comparison of both of them. So by definition, eukaryotic cells, so it would be any cell that contains a clearly defined nucleus and has membrane bound organelles basically. And our prokaryotic cells, they do not have a true nucleus and they do not have membrane bound organelles. So example of our eukaryotic cells are like our animals, plants, fungi, and protist cells. An example of prokaryotic cells would be like our bacteria and our archaea, right? So now nucleus is very much present in our eukaryotic cells, and then in our prokaryotic cells, the nucleus is very much absent. So cell size, if we're comparing the cell size, so our eukaryotic cells are a lot more larger than our prokaryotic cells, right? So they are between 10 to 100 micrometers, while our prokaryotic cells are a lot smaller, which means that they could be less than, they can be, let's say, a max of maybe 5 micrometers. They're really small. So, yeah. Anyway, so DNA replication highly regulated with selective origins and sequences for eukaryotic cells, and in our prokaryotic cells replicates entire genome at once. I always pronounce this word really wrongly. And then you have organism type. The eukaryotic cells, they're usually very much multicellular, right? So not all eukaryotic cells are multicellular, but most of them are. For our prokaryotic cells, they are strictly unicellular, like completely strictly unicellular, right? So eukaryotic cells have more than one chromosomes, they have large ribosomes, they have a slower growth rate, the organelles are very much present, ability to restore hereditary information. So they have a true nucleus, right? And they have a cell wall which is present in our plants and fungi. We have our cell wall being made up of cellulose in our plants, and our cell wall being made up of chitin in our fungi. And we very much have a plasma membrane, and plasma membrane is the same thing as a cell membrane. It's just another term for membrane, basically. The cell membrane, plasma membrane, it all means the same thing. And then the cytoplasm is very much present. Then over to our prokaryotic side. So yes, we have a small ribosome. So basically, eukaryotic cells have 80s ribosomes, and prokaryotic cells have 70s ribosomes, right? That's why they say it's smaller. Growth rate faster, organelles are absent, so the prokaryotes do not have membrane-bound organelles, and it says all prokaryotes have this ability to store hereditary information, right? So they don't have a nucleus, but they do have a nucleoid, and they also have a plasmid present, right? And the plasmid is like some free floating DNA in their cytoplasm, right? So the cell wall is a bit more complex. So in our prokaryotic cells, specifically our bacteria, the cell wall is made up of peptidoglycan. That is how I pronounce it, but the cell wall is made up of peptidoglycan, and then plasma membrane is also present, cytoplasm is also present. So finally we get to the cell. So basically this is just a rough draft of an animal cell, basically, right? So we're going to use this animal cell as the rubric for what we're trying to do today, which is talk about the structure and function. So we have our big nucleus, our nucleolus, which is inside, chromatins right here, and basically it's chromatin before it has entered the division stage, basically before it has undergone mitosis. Yeah, if it has undergone mitosis, that's what I mean to say. And then you have the rough endoplasmic reticulum, and if that's the rough endoplasmic reticulum, over here should be the smooth endoplasmic reticulum. It's not really differentiated, but yeah. Um, your nucleolus, ribosomes, Golgi apparatus, Golgi vesicle, cytoplasm, vacuole, lysosome, peroxisome, smooth endoplasm. So okay, so here it is, smooth endoplasmic reticulum. So this diagram is a bit different, but so rough and the smooth endoplasmic reticulum, and then the secretory vesicle, intermediate filament. So I guess this is the smooth endoplasmic reticulum. because what they usually do is they usually have like the entire endoplasmic reticulum, and then they would show the ribosomes on one side, and then over here would be like without ribosomes. But in this case, in this drawing, they decided to put the smooth endoplasmic reticulum kind of separate here. However, this is just a simple animal cell diagram which you should be able to draw for your exam and label on your own. So let's talk about the cell structure and function. So, cell structure and function. So our nucleus, which is a double membrane organelle, right? So our nucleus is actually surrounded by a nuclear envelope, right? And the function of our nucleus is to basically control the cell activities, right? So if they ask for the structure, you would tell them that it's a double membrane organelle surrounded by a nuclear envelope, right? So that is why it's double membrane, right? Because it's not just one membrane around it. It's surrounded by a nuclear envelope, and on this envelope there are some pores, and that's basically how the exchange of information between the cell and the nucleus takes place. Then we have our mitochondria, and as I said before, the first definition I ever learned about our mitochondria is that it was a powerhouse of the cell. Now let's do some differentiation between mitochondria and mitochondrion. So mitochondria is actually very much plural, right? So it means more than one, right? Which in our cells we do have more than one mitochondria. It's not like the nucleus where it's just one in the cell. In the case of the mitochondria, we have a lot of mitochondria all over the cell. So whenever you do talk or write about mitochondria, bear in mind that mitochondria is plural and mitochondrion is singular. So when you're talking about a single mitochondrion, you would say mitochondrion. If it's plural and you're talking about a lot of them, then you would say mitochondria. So the reason why it's called the powerhouse of the cell is because it produces ATP, and ATP is absolutely necessary for the cell to function. Without ATP, without energy, the cell has nothing going on. How would the cell carry out its cellular activities if there's no energy, right? So the mitochondrion. So in this situation, we said mitochondrion, we mean singular, so we're talking about one here. So it's also double membraned, right? Rod shaped. It's a double membrane rod shaped structure that is found in both plant and animal cells with cristae foldings. So this means that the mitochondrion, or the mitochondria, since we're talking about this, so the mitochondria is not just found in animal cells, it's in both animal and plant cells, right? It is there basically in all, I would say all eukaryotic cells basically. Because what's going on here is that within this cell there has to be a powerhouse, right? It is a powerhouse and it produces ATP, right? And it plays a big in a lot of functions that take place in the cell, right? So in our mitochondria, what we have is some okay, so this is it. Let's use this. So on this diagram, our mitochondria is actually not labeled, but this is our mitochondria. So it has an outer membrane and an inner membrane. And so with this mitochondria, it has some folding inside, like some foldings inside, which is called, we call these cristae, right? Cristae, and it's also a double membrane organelle, just like the nucleus. It's also double membrane with cristae inside in this situation. But there's an interesting story behind the mitochondria and also chloroplast, but I won't really get into that story, right? So it has a double membrane, just like it. And then let's talk about our chloroplasts. So they're only found in plant cells. So basically in this animal cell, you will not find any chloroplast whatsoever. No chloroplast. So basically it's only found in plant cells, in well at this level, contains a green pigment called chlorophyll. It carries out photosynthesis and it's also double membraned, and it contains some structures called thylakoids, and basically they're like some flat circular structures, right? And when they are stacked, they form a granum, and basically the space within the chloroplast would be our stroma, which it would be like the space slash cytoplasm of the chloroplast basically. So in this diagram we will not find a chloroplast, but there's an interesting story behind, well, it's a theory behind the mitochondria and the chloroplast, and basically it's the endosymbiont, or am I pronouncing it properly, endosymbiotic theory. And basically it's saying that the reason why the chloroplast and the mitochondria, why the reason why they have double membranes, and it's almost like they have, how could I explain it, they have some characteristics which makes some scientists believe that they were once original like cells outside of a eukaryotic cells, if that makes sense. So the theory is that they were actually like swallowed, they were actually a type of bacteria, and we know that bacteria is like unicellular. So the theory is that the mitochondria and the chloroplasts were once unicellular cells that were free living independently, and they got swallowed up by a cyanobacteria, so basically a bigger bacteria, and then that bacteria is what evolved to become a eukaryotic cell. That's a theory basically. Okay, so basically that's the theory behind the chloroplast and the mitochondria, that they were basically once bacteria. But anyways, let's continue. So ribosomes, we have 80S ribosomes in our eukaryotic cells, and we have 70S ribosomes in our prokaryotic cells. Now, we already mentioned this before earlier, but it does make sense that we have the larger ribosomes in eukaryotes, because after all, eukaryotes are basically a lot larger than prokaryotes. Basically, prokaryotes are so small, bacteria is so small, and if we should compare the size of a chloroplast or the size of the mitochondria, that would be the size of a prokaryote, basically the size of a bacteria. So if you look on it like that, the eukaryotic cell, a eukaryote or animal cell, if we use it as an example, it's basically 10 times bigger than a prokaryotic cell. So that is why the ribosomes are bigger in our eukaryotes. So 8 is ribosomes in our eukaryotes, 7 is ribosomes in our prokaryotes, and it makes sense. So basically the primary function is protein synthesis. So the ribosome now, the structure of the ribosome is actually made up of two subunits. You have a large one and you have a small one. Now, how I would describe the structure is that basically you have a, like, make a fist with like your thumb sticking out on one side, and then just fold up the other hand and kind of like put it on top, but just hide the other thumb. But basically that's how the ribosome structure kind of looks. So one of them large and one of them small, so the two subunits come together to make the ribosome. And as we said, 8 is ribosomes in our eukaryotes and 7 is ribosomes in our prokaryotes. Now the endoplasmic reticulum, you have two main types of ER within our cells. We have the smooth endoplasmic reticulum, and basically the smooth endoplasmic reticulum produces lipids and steroids. Then the rough endoplasmic reticulum is responsible for protein synthesis. Now, the reason why this makes sense, so if we go back to the diagram, actually, let's go back to the diagram. As you can see, the rough ER literally has ribosomes on it. You can see the little jukes up them. Those are ribosomes. Compare it to the smooth endoplasmic reticulum that don't have any of those stuff. So you have the ribosomes which are on the end of the rough endoplasmic reticulum. You have ribosomes that are free floating. See them here? They literally label it free floating ribosomes. So basically protein synthesis, and the structure of the endoplasmic reticulum is basically flattened sac-like sheets called cisternae. So in our ER we have cisternae, in our Golgi we have cisternae, in our mitochondria we have cristae basically. So that's the difference between the foldings. Okay, now to some other organelles. So basically our lysosomes. As you can see, the lysosome is really small. It's literally like a little circle. So basically, the main function of our lysosome is to break down waste materials within the cell. That's the main function of lysosomes. So basically, they have hydrolytic enzymes, and these hydrolytic enzymes are responsible for breaking down these waste substances. So lysosomes play a big role in clearing out the cell, clearing out the waste. It helps to break them down, right? And also the lysosome, so it's actually formed from the Golgi apparatus / the Golgi body. So that's where the lysosome is formed from. So it literally would get packaged or pushed out by the Golgi apparatus, which we soon talk about the Golgi. But that's what it does. It has hydrolytic enzymes that break down cellular waste and debris by engulfing it basically. Or cytoplasm is basically the site of the cell's metabolic activities. It's like that jelly-like, fluid-like substance that literally surround everything. All of the membrane-bound organelles literally sit in the cytoplasm. Peroxisome, these structures basically get rid of toxins within the cell. That's the main thing about our peroxisomes. They are spherical or ovoid shaped with a single membrane core containing many enzymes. Basically, but the main function of our peroxisome is to get rid of toxins within the cell. And of course, our cell membrane is the outer covering, controls what moves into and out of the cell basically. Now, let's talk about the Golgi. So earlier, I said, okay, the Golgi package the lysosomes and basically send them out, right? So the Golgi apparatus, as I said before, it has cisternae. So basically, these stacked, flattened, fluid-like sacs called cisternae, Golgi apparatus has it, and the endoplasmic reticulum has it as well. And the Golgi is the packaging center of the cell. So everything go to it, it package it, and then it distribute it where it needs to go afterwards, or vacuole. Now, in our animal cells, the vacuoles are really small compared to like a plant cell. Within a plant cell, you would see like a really big vacuole. Like the vacuole take up like, let me say, 3/4 of the cell in a plant cell, right? And we know why plants need such a big vacuole, right? Because they are involved in making their own food. They carry out photosynthesis. They have a lot more waste materials. They need a lot more water. So basically, the vacuole stores food and also removes waste. And we understand why a plant cell would have like such a big, a big vacuole versus an animal cell that has such a small vacuole just scattered all over the place, right? So it's not here, but let me just mention the cell wall. So basically in our plant cells, they have cell walls made up of cellulose, and basically it's that rigid structure that surrounds the cell, protects the cell. It's literally over the cell membrane. So our plant cells have cell walls, or animal cells don't. Now that means that animal cells can't burst. Like if you look at like some diagrams where, let's just say something happened, or the cell becomes turgid or something, right? And let's just say the cell bursts. That's an animal cell. Plant cells won't burst because of that cell wall, right? And the cell wall is what gives plant cells their fixed shape. So they're not like, circle or irregular like animal cell. They have a fixed shape basically. So that's really the function of the cell wall. And even our prokaryotes, specifically our bacteria has peptidoglycan in its cell wall, or fungus which we not really get into fungus, but fungus has chitin in their cell wall, and fungi, that would come under eukaryotic cells. The bacteria comes under prokaryotic cells. Now, it's also important to know the difference between a chromatin and a chromosome, because look at this diagram right here. We know this. So basically, one of this is the sister chromatid, and it's literally connected in the middle by a centromere. Now, what is the difference between them? So basically, let's go back to the diagram. So basically, you see in here, the diagram points to chromatin. So the main way that you can differentiate chromatin from the chromosome is that during so basically, you know the cell cycle, where you have interphase, and then you get into like mitosis or meiosis. But in this situation, so for example, the cell spends most of its time in interphase, right? So during that phase, the cell is not dividing, it's just there, it's just resting, it's just preparing for cell division, right? Now during that time, the chromosomes, keyword, this is to be carefully, okay, let me be careful with the wording. Basically, the chromosomes are not condensed, let me just say that. And that is why those chromosomes that are not condensed, those are the chromatins. That's before cell division, before mitosis start take place and all of that stuff. Because we know that once it enters into if let's use mitosis, once it gets into prophase is the stage where the chromosomes become condensed. So before that, they were just chromatin, right? They were just chromatin before cell division. They're basically like a lower class of, you know, DNA or whatever, right? During mitosis or cell division, however you want to term it, they become condensed in prophase, and when they become condensed in prophase and we can see them, that is when they become chromosomes. So basically, the DNA packaged by special proteins called histones to form chromatin. So this chromatin further condenses to form chromosome. So basically, the uncondensed version is the chromatin that's before cell division. The condensed version is basically during cell division, during mitosis, during prophase, when the chromosomes become condensed and they become visible. You can start to see them nuclear envelope breakdown and all them something here, right? So chromatin is not involved in cell division. Right? So it's in the cell cycle in interphase, just sit down and nod and nothing basically. Right? Like the cell is a prepare, but the cell is a prepare, rest, same thing, whatever, it's just there. When it enters into mitosis prophase and it become condensed, that's when it become a chromosome. So basically, this means that the chromatin is a lower order of DNA organization, whereas chromosomes are a higher order of DNA organization. So basically, that's the main difference between the two. And this is just like a summary diagram of the animal cell versus the plant cell again, which you can look on some more. And as you can see, the big vacuole versus let's see, this diagram that you've not showed the vacuole of an animal cell, but we know it's there, but them the small. But the point is that the plant cell has a really big one. You can see the cell wall, the cell membrane or chloroplast or nucleus or nucleolus inside. Alright, or mitochondria, all of that stuff. Right? So basically, I think that's the end of it. Oh yeah. So that's it. So the animal versus the plant cell, chromatin versus chromosomes, Golgi, vacuole, lysosome, cytoplasm, peroxisome, cell membrane, chloroplast, ribosomes, endoplasmic reticulum, nucleus, mitochondria. Diagram again. So basically, I think that's the end of it. That's the end of the cell structure and function, basically.